start the class. Today we are dealing some few important topics. The first thing we are dealing is hemochromatosis, which is also recently important question, which is usually asked from endocrinology department. Okay. Once this hemochromatosis is completed, we'll complete our endocrinology. Then we'll start GIT. In the GIT, we'll go for irritable bowel syndrome. And third, we'll deal for metabolic associated fatty liver disease. And with this, we'll complete GIT. Then we'll go to CNS. In CNS, we'll deal coma. And fifth is brain death. So today, if time permits, we'll cover all these 5 topics today. In this, the coma and the brain death, these last 3 topics are very, very important, which you cannot even difficult to write from the Harrison. So I will make it a simple thing. So first we'll complete the simple things and then we'll go for the coma and brain death, right? So let us start our discussions. We'll start with the hemochromatosis. So let us begin our discussion with the topic called hemochromatosis. So what do you understand with the term hemochromatosis? So hemochromatosis is nothing but actually it's an iron overloading condition. Actually it's a group of genetic disorders which are characterized with iron overloading. That means there's an iron overloading. This iron gets deposited in different tissues and all the tissues undergo organ damage and they manifest many symptoms. That is what is called as hemochromatosis. When you come to definition, it's a group of genetic diseases characterized by iron overload leading to organ damage. So group of genetic disorders characterized by iron overload leading to the organ damage. This is what is called as hemochromatosis. Now why this iron overload happens? So this iron overload, it can happen due to two reasons. Either it can be primary or it can be secondary, right? So primary is mainly it is genetic conditions and secondary is because of mainly from secondary causes which cause iron to accumulate in the body. So before going to primary, just let us see what are the different secondary causes which can cause iron overload. If it is secondary conditions, so mainly one because of anemia. So one because of the anemia. So if there is any chronic anemia, then what will happen in order to form red blood cells, our body tries to absorb more amount of iron from the gut. So it leads to what is called as iron overload. And also we try to transfuse the blood to the patient. That also leads to what is called as iron overload. So anemia conditions are mainly thalassemia, any sideroblastic anemia, and anemia due to any chronic hemolytic anemia. So any anemia is there, actually there's a more amount of iron absorption from the gut that leads to what is called as iron overload. And second thing is increased iron intake. So usually nutrition will give increased iron intake. Some people whenever they feel generalized weakness and all, they will take iron tablets and all, and some patient RMPs and all, they used to prefer to give iron injections. So increased iron intake. And third is any liver disorders. So any liver disorders can also cause this thing. It can be alcohol, or it can be hepatitis C virus, or it can be your non-alcoholic fatty liver disease. Anything they can cause iron overload. So these are the secondary conditions which is causing your iron overload, right? So just I'm repeating, the hemochromatosis is a group of genetic disorders characterized with iron overload and leading to organ damage. This iron overload can be primary or secondary. The primary is genetic and secondary are because of due to anemia or due to liver disorder or due to increased iron intake, right? If this is done, I'll just go for the primary iron overload. Primary cause of iron overload. They are mostly genetic conditions. So before going to know this thing, I just want to let you know a basic detail about iron absorption, how is it happening and what are the genes involved in this iron absorption, so that we can know about hemochromatosis also, right? For example, now this is what we call a gut. This is your enterocyte. This is the lumen. Okay, so whatever you're eating, the iron will come into the gut. It converts to Fe2 plus. Now this Fe2 plus enters into the enterocyte by means of what is called as divalent metal transporter 1. Okay, because of divalent metal transporter 1, this Fe2 plus is carried into the enterocyte, and now this Fe2 plus will reach into the circulation through the action of one protein called as ferroportin. So this ferroportin is the one protein which helps in transfer of iron from the enterocyte into the blood vessels, right? Now, in the blood, this iron in the ferroportin is carried by the transferrin. So transferrin carries this iron and it takes to what is called as your bone marrow. Okay, in the bone marrow it is used by the erythrocytes to form hemoglobin and all, right? This is actually normal iron that is taken in the diet and all. It goes to lumen, intestinal lumen. In the intestinal lumen, it is carried to the enterocyte by action of divalent metal transporter 1. From the enterocyte, it reaches the blood circulation by action of ferroportin, and now in ferroportin, it is carried in the circulation by using a protein called transferrin. From this transferrin, this iron is taken to the bone marrow, where RBC is taking this iron and helps to synthesize hemoglobin. Clear? Now, whenever there is actually in the bone marrow, RBCs, whenever they are dying, they are senescent. Then again, this iron is going to transferrin. Right? Right. Now, what is happening in the liver? We'll see now. So, this is what is called as liver. So, liver, whatever the excess iron is there, it is stored in the form of ferritin. This is all storage form of iron. Right? So, this liver, what is called, it synthesizes a protein called as hepcidin. Very, very important, hepcidin. Okay. This liver synthesizes a protein called as hepcidin. This hepcidin has inhibitory effects on two things. This hepcidin inhibits this ferroportin. So thereby it inhibits the absorption of iron from the gut. Similarly, hepcidin has inhibitory effect on bone marrow or the macrophages, so that it inhibits the release of iron from the senescent RBCs. Clear? Any doubts? Okay. I'll just come again, don't worry. So actually, the iron that is taken from the diet, it is going into the intestinal lumen. From the intestinal lumen, it goes to enterocyte by action of a protein called as divalent metal transporter 1. From the enterocyte, it enters into circulation by action of a protein called as ferroportin. Right? In the circulation, it is circulated through transferrin. From the through transferrin, it goes to bone marrow, where it is taken by the RBC and helps in synthesis of hemoglobin. Whenever the RBC dies, again the excess iron is going to the transferrin. Whatever the excess iron is present, that is stored in the liver as hepcidin, stored in the liver as ferritin, and liver synthesizes one important protein called as hepcidin. This hepcidin has two important functions. One, it inhibits the ferroportin, thereby inhibiting the absorption of excess iron. Similarly, it also inhibits the release of iron from the bone marrow and macrophages. So this liver synthesizes hepcidin only whenever there is iron is excess in the body, then liver will synthesize hepcidin. So this hepcidin inhibits the absorption of iron and release of iron from the bone marrow, so thereby preventing iron overload in the body. Clear? So whenever this hepcidin is lost, there is no inhibitory effect on the gut and there is no inhibitory effect on the bone marrow. More amount of iron is coming into the body, and that leads to what is called as iron overload. Clear? Now, what are the things that can affect the hepcidin? So hepcidin, it is synthesized by the liver mainly to decrease iron absorption from the gut and also to decrease iron release from the bone marrow, thereby preventing your iron overload. This is a protective mechanism which our liver develops by synthesizing hepcidin. Right? So basically, whenever you see this thing, this hepcidin is stimulated by two things. One is your transferrin levels, that iron that is bound to the transferrin levels, that is nothing but your serum iron. So circulate in a circulation, how much iron is there, how much iron is bound to transferrin levels, that will determine your hepcidin levels. And second thing is, the thing which determines hepcidin is liver iron stores. Okay, let us see. So for example, this is your liver cell. Okay, so whenever there is circulation, this is what is called as blood. This is your blood. In this blood, iron is present in the form of what is called as transferrin. Okay, there is a single gene called as HFP gene, homeostatic iron regulator gene. Right? So this the protein is a HFP protein. Actually, this is a HFP protein. it will sense the iron that is present in the blood in the form of transferrin. Then it will interact with what is called as transferrin receptor 1, right? Based on synthesis, whenever this iron is low, then it will stimulate the DNA and helps in synthesis of what is called as hepcidin. Okay, so whenever serum iron is low, that is sensed by this homeostatic iron regulator protein, which correlates with transferrin receptor 1 and stimulates DNA and synthesizes hepcidin. This hepcidin helps in absorb. Whenever iron is excess, sorry, Madhu. So whenever the iron is excess, then this HFE is stimulated and synthesis leads to synthesis of hepcidin. This hepcidin inhibits the absorption of iron. Clear? Similarly, one more stimulus factor is this liver iron stores. Okay, whenever this liver has excessive iron stores are there, then one more protein is called as hemojuvelin. It will get stimulated and then leads to what is called as some bone morphogenic protein SMAD pathway is involved and also leads to what is called synthesis of hepcidin. Right? So in liver, what is happening? There are two things are there. Whenever this liver needs to synthesize hepcidin, and that is determined by two levels. One is your circulatory iron levels, and second is liver iron store. Right? Whenever there is circulatory iron levels are more, whenever the blood circulation has more iron levels, your homeostatic iron regulator protein is stimulated. It will interact with the transferrin receptor 1, and then it stimulates the DNA, and more amount of hepcidin is produced, so inhibits the absorption of iron. Clear? Similarly, whenever the liver iron stores are more, then hemojuvelin protein is stimulated. It goes through bone marrow protein and SMAD pathway. Hepcidin is also increased. So these two pathways are involved. So any defect in these pathways or any defect in these genes or any defect in these proteins leads to decreased sense of hepcidin, then more absorption of iron. So iron overload, and that is your hemochromatosis primary. Clear? Any doubt is there? Just raise the answer. We can explain again. There is only difficult thing in hemochromatosis. If it is understood, everything is understood. Can I go further? Okay, fine. Now let us see. So simple thing. So there are two genes are there. One is called as homeostatic iron regulator. Other is called hemojuvelin. And there is a transferrin receptor 1 protein. So anything defect in these things, there is no synthesis of hepcidin. Then there is iron overload is present. Okay. So now, whenever there is defect in what is called as homeostatic iron regulator gene, HFE gene, that is what is called as hemochromatosis type 1. Okay, this is the most predominant. So that's it's called as HFE gene related hemochromatosis. There are other conditions called as non-HFE gene related hemochromatosis. Okay, so what other genes are called? Hemojuvelin. If hemojuvelin mutation is there, that is called as juvenile hemochromatosis, which is seen in mostly in childhood. That is a type 2A. Okay, clear? And if there is any defect in the hepcidin, that is what is called as juvenile hemochromatosis, that is type 2B. Okay, or if there is a mutation in ferroportin. What is ferroportin? It's present on the basolateral membrane of enterocyte, which helps in absorption of iron from the enterocyte into circulation. If there is a defect in enterocyte, mutation in the ferroportin, that is called as type 3 hemochromatosis. Or if there is mutation in transferrin receptor 2 protein, that is a type 4 hemochromatosis. Clear? Just a small change. This ferroportin is type 4 and transferrin receptor 2 is type 3. Okay. So if this is done, then we can go very easily. I just want to repeat this thing once. Okay. So actually, so simply speaking, I'm saying just the main cause of primary overload is actually the iron that is taken by the body, that is taken by diet. It enters into the lumen of the gut. From the gut, it enters into the enterocyte by action of NM diviral metal transporter 1 protein. right? From the enterocyte it enters into circulation by action of protein called ferroportin, right? Now from in the circulation it transfers in form of transferrin. It is taken by the bone marrow, helps in sense of the hemoglobin. Again RBC, whenever it dies, whenever its senescent is there, then all this iron is getting transferred to in the form transferrin, right? Whatever the excess of iron is there, it is stored in the liver as ferritin. In the liver there is a protein called as hepcidin. This protein inhibits absorption of iron from the gut and also inhibits release of iron from the bone marrow, thus preventing iron overload. Now what are the things that are stimulating this hepcidin? So two things, one is transferrin levels, other is your liver iron stores. Whenever the serum transferrin levels are more, then there is a gene called as HFP gene, homeostatic iron regulator gene. It senses a protein, it interacts with the transferrin receptor 1, then it stimulates the of hepcidin. Similarly, whenever your liver iron stores are more, there is another protein called hemoglobin. Then through one pathway, it also releases of hepcidin. Then it inhibits the absorption of iron. So any of these proteins are defect is there, then there is no synthesis of hepcidin. Then more amount of absorption of iron overload can happen. Okay, if it is mutation of the protein of homeostatic iron regulator gene, that is called as type 1 hemochromatosis, and other things are called as non-HFE gene related. In this, hemoglobin is type 2A, hepcidin type 2B, transferrin receptor 2 is type 3, and ferroportin is type 4. Clear? Right. Yeah. Where is TFR2 located? The same thing. This is TFR1 and TFR2 both will be there, right? 1 or 2, right? Clear? So this thing, just a simple chart I can make for you now. So this one, when coming to hemochromatosis, this can be primary or this can be secondary, right? In the case of primary, one is homeostatic iron regulator gene related, other is non-homeostatic iron regulator gene related, right? This is called as your type 1. In this you have type 2A, type 2B, type 3 and type 4. This is hemoglobin, this is your hepcidin, this is your TFR2, this is your ferroportin, right? Clear. And then in the secondary, you have either due to anemia or either due to increased intake or due to your liver disorders. Clear? These are hemochromatosis, primary, secondary. We can divide this classification like this. So you can study like this. You can even write this classification exam like this one. So this is the basic concept for this hemochromatosis. Right. Now, so now we understood what is definition of hemochromatosis and how iron overload is happening. So what are the main regulator proteins hepcidin and what are the things that are regulating hepcidin we came to know. Now because of decreasing hepcidin, there is more amount of iron overload. Iron is getting absorbed in the body. So if iron is getting absorbed in the body, what is going to happen? So when you see the clinical features or the course of the disease, if you see this thing, you can see one thing. Actually, the course can be categorized 4 types. One, actually, people who have genetic mutation present but no iron overload. This is one spectrum of people. Okay. Second spectrum is iron overload is there present but no symptoms. Okay. And third spectrum is symptoms are present due to iron overload but no organ damage. And fourth spectrum is you have organ damage. So this is a clinical course of hemochromatosis. So this is, they go into 4 stages. First is actually there is genetic mutation only, there is no iron overload. Second, there is iron overload but there is no symptoms. Third, there is symptoms but there is no organ damage. And fourth, there is organ damage. Clear?